Hi students, how are you? I hope each one among you are doing great and well. Welcome to Supnit's classes. This is Rahmat Latif, your biology educator. So in this topic, we are going to learn female reproductive structure. Yes, you heard me correct, female reproductive structure. First of all, we will see what does female reproductive structure is consisted of. As I already mentioned, it's consisted of pistil and it has megasporangium and embryo sac. Okay, see, here in female reproductive structure we have pistil, but in male reproductive structure we had stamen. Here we are having megasporangium, there we had microsporangium. Here we are having embryo sac, there we had pollen grain. That means, as I already mentioned, that pollen grain is not male gamete. Pollen grain is male gametophyte. I am repeating. That means it's carrying male gamete. Pollen grain is male gametophyte. That means it's carrying male gamete. And here embryo sac is female gametophyte. Okay, female gametophyte. I hope this concept is clear. That means inside embryo sac we have female gametes. Okay, great. I'm erasing it. Now in a flower we may have single pistil or we have so many pistils, right? Wait, let me erase it. In pistil we may have single pistil or we may have, in a flower we may have single pistil, okay, and that is known as monocarpellary. And we may have so many pistils, multiple pistils. That is known as multicarpellary. Okay, these carpels, these multiple carpels may be fused together, and that is known as syncarpus, in which we have carpels in fused form. Okay, or we may have pistils in separate form, not fused, they may be free, and that is known as apocarpus. I hope this concept is also clear. What is monocarpellary and what is multicarpellary? Okay. Okay, moving ahead. See, I'm again forming the structure of pistil. Let's recall the structures which are present in, flower, female reproductive structure. Say, for example, this is upper portion is stigma, then we have style, then finally we have ovary, bulged portion, okay, like this. This is stigma, this is style, this is ovary. Okay, here inside ovary we have ovarian cavity locule. Here we have placenta. What? We have placenta. Okay, arising from placenta we have a structure like this. This is known as funicle. Okay, it gives attachment between ovule and placenta. And in ovule and funicle we have one more structure, that structure is like this, and that is known as hilum. Okay, that structure is known as hilum. And finally we have a structure like this, which is known as ovule, which is present inside ovarian cavity. Okay, ovule gets converted into seed, ovary gets converted into fruit. Okay, I hope this concept is clear. Please remember this is the question of PYQs. What's the structure that is present between ovule and funicle? That is hilum. And if they may ask you what's the structure present in between, um, um, ovary and ovarian cavity, that is placenta. Okay. I hope this concept is clear. And this ovule is also known as megasporangium. That means inside ovule we will get embryo sac will get developed, and embryo will get developed. Fertilization will take place. Okay. I hope this concept is clear. Let's move forward. We will just draw this ovule now. Okay. I'll show you what is inside ovule. This is our ovule. Okay. This is ovule like this. And ovule has, it has two coverings. This is first covering, blue one. Okay, this is first covering, and this is another covering. Sorry, this is another covering. Okay, like this. Inside this red structure is, let me join it in order to make it more clear. This red is what? It's ovule. This blue one and white one are integuments, coverings of, ovule. This blue one is inner covering, inner integument, and white one is outer integument. Okay, and inside ovule we have, so many tissue present, a lot of tissue present. That tissue is known as nucellus. And these coverings, either in inner covering or outer covering, they just cover whole nucleus except at one end, right? This end, except at this end. Okay, except at this end. And this end is known as micropylar end. It's like small structure end, right? That's why it's known as micropylar end. Okay, I hope this concept of ovule, structure of ovule is clear, right? Okay, and this is, since this is micropylar end, the structure opposite to micropylar end, this site is known as chalazal end or calazal end. Okay, this is micropylar end, and the structure and the site which is present opposite to micropylar end is known as chalazal end. Okay, very nice. Now please see. Here at this end we have a large cell that is known as megaspore mother cell. Okay, megaspore mother cell, MMC, and it gets converted into embryo then. Okay, what's the function of nucellus? What's the function of nucellus? Its function is to, it just act as a reservoir of food material. It provides nourishment. Okay, it provides nourishment to the developing embryo, or we can say to the megaspore mother cell. Now we will see, as we saw in microspore mother cell, what happened to microspore mother cell, we will see in megaspore mother cell as well. Here is megaspore mother cell. Its polarity is also 2n, same as microspore mother cell. Then what happened in microspore mother cell in male reproductive structure? That undergoes meiosis 1. It will also undergo meiosis 1, and it will form a structure like this, N and N. These cells will also undergo meiosis 2. Okay, and after meiosis 2 they will form 4n like this, 1 2 3 and 4, right? Same as what they as which was formed in, male reproductive structures, microspore, that is known as microspore tetrad. and therefore give rise to pollen grains, right? But here what happened? Three of them degenerate. They degenerate. Three of them degenerate. And what will happen to this one? We will see on our next slide. This is megaspore, right? What will happen to this megaspore now? See, it will undergo mitosis. Okay. After mitosis, it will form two, nucleus, not cells, because cytokinesis won't take place. It will form, it will undergo continuously as karyokinesis, but not cytokinesis. Okay. Like cytoplasm will not be laid on the cells. They would be in one cell forming more than one nucleus. Okay. After one mitosis, they will form two nucleus, like 1 and 2. Then it undergoes another mitosis. It will form like this: 1, 2, 3, 4. It will undergo another mitosis. It will form like this: 1, 2, 3, 4 will give 8, right? 1, 2, 3, 6, 7, 8, right? 1, 2, 3, 4, 5, 6, 7, 8. That means 8 nucleus are now in single cell. 8 nucleus are in single cell, right? See, now after this stage, what will happen? After this stage, they will undergo what? Cell cytoplasm, cytokinesis. Now cytokinesis will occur. I'll show you how. I'm going to erase this. Now we will see what's the arrangement of, nucleuses in this cell and which cells will be formed. Okay. See what will happen. This is cell. Sorry. The cell was formed, right? This cell was formed, and we were having total 8 cells: 1, 2, 3, 4, 5, 6, 7, 8 cells, right? See what will happen inside this cell would be formed like this. And what will happen? 3 cells will get formed here: 1, 2, and 3. Then we have 3 cells here like this: 1, 2, and 3. Here we have 2 nucleuses. Inside them we have their own nucleus. Now please count. We were having 8 nucleus. 1, 2, 3, 4, 5, 6, 7, 8. Here we are having how many nucleus? 1, 2, 3, 4, 5, 6, 7, 8, right? 8 nucleus in total. Now cytokinesis occur to these 3 cells. 1, 2, 3. They will get converted into cell. These 3 also got converted into cell. 1, 2, 3, right? That means in total we have 8 nucleus, right? In total, we have 8 nucleus, but how many cells? This one cell, 2 cell, 3 cell, right? Then 4, then 5, then 6, and we have these 2 nucleus in a single cell. Okay, that means we are having in total 8 nucleus, 7 cell, right? 7 cell. And this kind of formation is known as 7-celled 8 nucleated structure. Okay. This is known as 7-celled 8 nucleated structure. I hope this concept is clear. How they are formed. How they are 8 nucleus but 7-celled. You got that. How they are 8 nucleus. But how only 7 cells? Why not 8? Because these two nucleuses here, they are in a single cell, right? Now we will just give them name. Okay, what are their names? These 3 cells at chalazal end are known as antipodals. Okay. These 2 cells at micropylar end, these 2 cells 1 and 2, it's known as synergids. These cells are known as synergids, and this cell is known as egg cell. Okay, this cell is known as egg cell. And here at synergids we have present finger-like structures, and these finger-like structures are known as filiform apparatus. Okay, these two nucleuses present in the center of the cell is known as, they are known as polar nuclei. Polar nuclei. Okay, and in which cell? Central cell. Okay, this egg cell and synergid, they together form egg apparatus. They are known as egg apparatus. Okay, apparatus. I hope this concept of female reproductive structure, how megasporogenesis took place, is clear. I am going to repeat. Say, for example, this is a cell, right? This is a cell. And what happens? This is a megaspore, right? Three of them degenerate. One is functional. And what will happen to that? Mitosis will take place, and it will finally form structure. That structure is known as seven-celled, eight-nucleated structure. And that structure at its callosal end contains antipodal cells. At micropylar end contains synergids and egg cell, which together form an apparatus known as egg apparatus. And in between the central cell, we have two nucleuses present. They are known as polar nuclei, right? And in synergids, we have finger-like structures. Those finger-like structures are known as filiform apparatus. Now, as we did in male reproductive structure, how many meiosis and mitosis took place in order to form male gamete? The answer was one meiosis, two mitosis. But here, what is the answer? How many meiosis and mitosis will take place in order to form female gamete? Okay, what is the answer? The answer is one meiosis, right? This, I'm again repeating, it's not different, separate meiosis. It's this meiosis contains two meiosis, meiosis 1 and meiosis 2. Okay, meiosis 1 and meiosis 2, that means single meiosis will take place. But how many mitosis will took place? 3. 1, 2 and 3 mitosis will took place. Okay, I'm erasing it. So in order to form female gamete, embryo sac. Embryo sac is female gametophyte, right? In order to form embryo sac, one meiosis and 3 mitosis took place. Okay, I'm going to write it down. In order to make female gametophyte, that is embryo sac, right? Embryo sac, one meiosis and 3 mitosis will take place. But what happened in order to form male gametophyte, that is pollen grain? One meiosis, same meiosis one meiosis will take place, but only 2 mitosis. But here, in order to form female gametophyte embryo sac, one meiosis and 3 mitosis will take place. I hope this concept of female reproductive structure is clear. Yes. Okay. So the mature, embryo sac, mature angiospermic embryo sac, that contains 8 nucleus and 7 cells. That's why we call the structure as 7-celled, 8 nucleated. Please remember this. Female gametophyte embryo sac is 7-celled, 8 nucleated. Only 7 cells, but 8 nucleuses. Okay, and I explained why, right? I'll write it down again because it's really very important. Female gametophyte, that is embryo sac, contains 7 cells and 8 nucleus. Okay, that's why we call the structure as 7-celled, 8 nucleated. I hope this concept of female reproductive structure is clear. Okay, we will meet in such any other session. Till then, goodbye, take care, thank you so much.